We start presenting. Okay, what I want to do is I want to review both chapters that are going to be on exam number 1. And then we'll talk about the other lectures that we did that are already on the YouTube channel. So I want to emphasize that 3 of you have not turned in your homework. Your PowerPoint presentations were due at noon today. I want you to know that that the ones that haven't turned them in, as of right now, will get some points deducted. So 3 of you that haven't turned them in, please turn them in.
Wait, they were due at noon?
Yes, they were. 11:59 a.m.
Oh. I did not know it was the AM.
Yeah, a.m. Okay? So get it in. All right?
Yeah.
Who's talking? I can't see who's talking.
Isaiah.
Okay. Isaiah, I won't take off, but just get it in, okay?
I'll get it in by today. I just didn't know it was noon and not like midnight.
No problem. That's fine. Don't worry. Okay.
I apologize. I didn't know it was noon either.
And you are?
Hannah.
Hannah, no problem. So Hannah and Isaiah, I won't take off any points, but get it in because I have to make a decision on the presentations, okay?
Okay. I'll turn it
All
in.
right.
Thank you.
Okay. All right. So there's only really one missing then because I know I've already gotten those 2. So there's only one missing. All right. So I'm going to re-emphasize what is going to be on exam number 1. I can tell you right now that these chapter lectures, 19 and 22, is 85% of your exam. 5% of your exam is going to be PowerPoint presentations, which are going to be given on Thursday. The other 10% is the supplemental lectures that I gave on HIV/AIDS, clotting, macrocytic anemias, blood typing. Okay, does that make sense to everybody? Somebody say yes or no.
Yeah.
Okay, good. All right, okay. So let me re-emphasize what you need to know. You need to know this slide. You need to know that when a tube of blood is centrifuged down, it divides into 55% plasma, 45% red blood cells, and in the middle is a buffy coat, and that contains white cells and platelets. You need to know this slide. This is a great slide because it really goes through everything. It tells you that the majority of your blood is basically plasma. Basically plasma. 55% plasma, 45% elements, formed elements. Out of the plasma, 7% is proteins, and here are the proteins, and they're very important proteins. The most abundant protein is albumin. Why is albumin important? Because it controls your blood pressure and your blood volume. Go back. Globulins, why are they important? Because of the immune system. Fibrinogen, why is that important? Because of clotting factors. Clotting factors. The majority of plasma is water. The formed elements. The most abundant formed element are red blood cells, millions of them, millions of them. White blood cells, they differentiate into neutrophils, lymphocytes, monocytes, eosinophils, and basophils. From the most abundant to the least abundant, never let monkeys eat bananas. Platelets, they are not cells, they are fragments of cells. Know this slide. On exam number 1, there are no pictures. Know the function of blood. There it is, right there. And the big one is it transports gases. It regulates homeostasis. It protects against excessive loss by clotting. And it fights infection. Here is a summary. We talked about this. This is a summary slide. You should definitely know this summary slide, okay? It says what I have already said, but it's important that you understand it. Lymphocytes can last a long time. Most white cells usually last only for a few hours, a few days, or a few weeks. Okay? The process of producing all blood cells, white cells, platelets, and red cells is called hematopoiesis. They all come from a stem cell. And this slide is important. Please know this slide. I ask a few questions on this slide. You basically have two stem cells that come from a bigger stem cell. This is called pluripotent, which means it can turn into anything. But basically, you have a myeloid stem cell and a lymphoid stem cell. The myeloid stem cell gives you everything but lymphocytes. The myeloid stem cell will even give you erythrocytes. Here's our erythrocyte, the red blood cell. It comes from a pro-erythroblast, which turns into a reticulocyte, an erythrocyte with a nucleus. Once the nucleus is ejected, that red blood cell now has only one function: transportation of nutrients and gases, hormones, etc. This myeloid stem cell can become a megakaryoblast. The megakaryoblast differentiates into a megakaryocyte, and that megakaryocyte breaks up into fragments called platelets, otherwise known as thrombocytes, and is involved in clotting. The myeloid stem cell can become an eosinophil myeloblast, which will become an eosinophil. It can become a basophilic myoblast, which will become a basophil. It can become a myoblast, which will become a neutrophil, a monoblast, which will become a monocyte. And when you talk about macrophages, macrophages are big eaters. Macro meaning big, phage meaning eat. They are neutrophils and monocytes. Neutrophils is the first macrophage to the site of inflammation. It goes away in 3 days, and monocytes stay for a month or two. Macrophages. The granular leukocytes are eosinophils, basophils, and neutrophils. The agranula are monocytes and lymphocytes. But lymphocytes come from a different stem cell. They come from the lymphoid stem cell. A T lymphoblast produces a T lymphocyte. A B lymphoblast produces a B lymphocyte. A natural killer lymphoblast becomes a natural killer cell. Please understand that an NK lymphoblast is part of the immunity that you're born with. This is non-specific. We all have it. We all have it. However, the T lymphocyte and B lymphocytes are part of the adaptive immune system, and they are very specific. The adaptive immune system has two important characteristics that differentiate it from the innate immune system. It's called immunity and specificity. I'm sorry, memory. I meant memory. Immunity, memory, and specificity. That's part of your adaptive immune system. Before I leave this slide, are there any questions on this slide? I'll take that as a no. Hemoglobin. Hemoglobin is the protein that is in red blood cells that carry oxygen and carbon dioxide. The heme molecule, which contains iron, is what carries the oxygen. The globin, which is the protein, carries carbon dioxide. Here it is. Hemoglobin. The iron is heme. The globulin are the polypeptide chains. Iron, the core of hemoglobin. Here it is. The core of heme. That's what carries the oxygen. That's what carries the oxygen. Okay? There's a formula in biology. It's known as H2 plus CO2 equals H2CO3. It is one of the formulas that determines the pH of the blood. All red blood cells contain the enzyme carbonic anhydrase, which converts CO2 plus H2O2 plus H2O into H2CO3. Water plus carbon dioxide equals carbonic acid. It also is the enzyme that reverses the formula, turning carbonic acid into carbon dioxide and water. You will learn in the respiratory system that bicarbonate, also known as carbonic acid when there is another H on it, transports 70% of the carbon dioxide in the plasma. So even though hemoglobin transport carbon dioxide, it's minimal. It's minimal. Carbon dioxide is transported in the plasma as carbonic acid or bicarbonate without another hydronium ion. The life of the red blood cell, know it, 120 days, period, know it. So now we talk about the breakdown of red blood cells. What happens when they die? Well, when they die, the phagocytes destroy the cell. It breaks it up into heme and globin. The globin, which is a protein, breaks down into amino acids. These amino acids are reused in protein synthesis. The heme, which is the iron part of the hemoglobin molecule, will turn into biliverdin and bilirubin, and the colors are here, green and yellow. Bilirubin will go into the liver and get stored in the gallbladder. Part of the heme molecule, which is the iron itself, will be transferred by a transfer protein called transferrin into the liver to make ferritin. Ferritin will now become iron, and transferrin will take it into the bones, into the bone marrow. Okay, so now you have iron and globin that's brought in through amino acids with B12 and folic acid. We talked about vitamin B12 and folic acid when I gave you the 3 presentations on macrocytic anemia. Macrocytic anemia. And because of the hormone erythropoietin produced by the kidneys, stimulates the bone marrow in the presence of hypoxia to produce more red cells. The liver takes bilirubin and stores it in the gallbladder. The gallbladder empties the bilirubin into the small intestine, and it comes out as feces. That's what makes feces brown. If it goes into the kidneys, it goes in as urobilinogen and becomes urobilin, which is yellow, and that's what makes urine yellow. That is what happens over 120 days to your dead red blood cells. Erythropoiesis is the production of red cells in the bone marrow. It is governed by erythropoietin. Erythropoietin is a hormone released by the kidneys in response to hypoxia that stimulates the stem cells to become erythroblast, which then become reticulocytes, which then become erythrocytes. And reticulocytes are immature red blood cells with a nucleus, okay? And the reason that's important is because if someone is anemic, has a low red blood count, they should have reticulocytes in their bloodstream, because with anemia, your tissues develop hypoxia, stimulating the kidneys to produce erythropoietin, EPO. And when you have stimulation of the bone marrow to produce more red blood cells, there are more immature red blood cells in the bloodstream. So that's how we know as doctors and medical professionals that your bone marrow is working. If you have an anemia and we don't see any reticulocytes in there, you've got a problem. And the problem could be your kidneys, You will learn in the renal system that when you have kidney failure, you become anemic. Why? Because your kidneys no longer produce erythropoietin and cannot stimulate your bone marrow to produce red blood cells. Any questions on red blood cells? All right.
Nope, all good.
Okay, good. Thank you for at least saying no. I appreciate it. Okay, leukocytes, white blood cells. They are a true cell. They contain a nucleus, they contain organelles, and obviously have no hemoglobin. Okay, they're classified as granular or agranular, and the granular lymphocytes are neutrophils, neutral granules. That means they have both blue and red. Eosinophils, only red granules. Basophils, only blue granules. Agranular, no granules. They are lymphocytes and monocytes. Okay, and there's what they look like. Obviously, I'm not going to show you any pictures on exam number 1. White blood cells can last for months. Most of them only last for a week or so. Okay, but during an invasion or inflammation, these white cells leave the bloodstream and they collect at sites of inflammation. And this is called emigration. Another word for that is diapedesis. They leave the bloodstream and they get into the tissue. And this is just showing you that they squeeze out of the bloodstream and get into the tissue. Okay? In general, an elevation of white blood cells indicates inflammation or infection. A low white blood cell count really does not mean too much, okay? But what's important is the differential white blood cell count. When you look at neutrophils, lymphocytes, eosinophils, monocytes, and basophils, okay? Because that can tell you what's happening with white blood cells, all right? This is an important slide. It tells you what happens when these white blood cells are high. I don't really care if you know about low. I want you to know high, all right? Neutrophils are more plentive when there is a bacterial infection or any kind of inflammation. Lymphocytes are more plentiful when there are viral infections, viral infections, okay? Monocytes are more plentiful when there are viral or fungal infections. Eosinophils are more plentiful when there are allergic reactions or parasites or autoimmune diseases. Basophils are much more plentiful when there are allergic reactions. I will tell you that the granules in basophils contain histamine. So do the granules in eosinophils. And in histamine is the primary problem in allergic reactions. That's why you take antihistamines, to get rid of histamine produced by basophils and eosinophils. Megakaryocytes are the mother of platelets. Megakaryocytes are cells that break into fragments, and those fragments are called platelets. The lifespan of a platelet is 7 days. 7 days. And here is another hormone, thrombopoietin. Thrombopoietin will make the pluripotent stem cell and the myeloid stem cell differentiate into megakaryoblast, which become megakaryocytes, which become platelets. Another name for a platelet is a thrombocyte, and they have a lifespan of 7 days. All right? Another review slide. You don't need to know the number of red blood cells or the number of white blood cells, but you need to know that neutrophils are the most common. Remember, never let monkeys eat bananas, from abundant to least abundant. Don't worry about bone marrows. That's really not that important. But you should certainly worry about clotting. And this clotting is known as hemostasis, the sequence of responses that stop bleeding. And the process includes, first, the vasculature goes into spasm. Then there's a platelet plug formation, and then there is blood clotting, or otherwise known as coagulation. Platelet plug formation and then coagulation. Coagulation is important because it is the presence of fibrin threads. And I gave you a lecture on the clotting cascade. I gave you a lecture that fibrinogen, under the influence of the enzyme thrombin, becomes fibrin. So fibrinogen is the clotting protein in the plasma. To reverse it, you subject fibrin to an enzyme called plasmin, and that enzyme breaks down fibrin into fibrinogen split products. Okay? And there is an intrinsic pathway and an intrinsic pathway. And here, we talked about this already. The extrinsic pathway is in the tissue. The intrinsic pathway is in the blood. This is the clotting mineral, calcium. Without calcium, you cannot clot. You cannot clot. This is the intrinsic pathway. This is the intrinsic pathway. Both pathways involve factor 10. Factor 10. They meet at the enzyme prothrombinase, which will turn into prothrombin, which will turn into thrombin, but where they meet is called the common pathway. Common pathway. And there it is. Fibrinogen becomes fibrin because of the enzyme thrombin. You should know fibrinogen. You should know calcium. You should know factor 10 because it's shared by both pathways. And you will know factor 8 because there will be a lecture on it on Thursday by one of your classmates. All of these coagulation factors are produced in the liver. In the liver. The vitamin that is used in clotting is vitamin K. Without vitamin K, you cannot clot. Without the mineral calcium, you cannot clot. To dissolve the clot, again, is plasmin. That's how you dissolve the clot. All right? Blood types. We talked about blood types. I gave you a separate lecture on blood types. And it's the ABO system. The ABO system. The most common blood type is O. Okay? And basically, they are antibodies, and these antibodies are known as agglutinins. And these antibodies can react with A or B antigens. And here is a slide that you have to know. When we give somebody blood, we give them packed red blood cells. We don't give them plasma. We give them red cells. Red cells. And the red cells have antigens on them, which will make your blood clot. Okay? So the universal donor is O, because you can give O blood to any blood type because it has no antigens on it. It has no antigens on it. The universal recipient, which can receive anybody's blood type, is type AB. So if you're type AB and you're bleeding to death, I can give you anybody's blood. because it has both antigens on it. Okay? Hemolytic disease of the newborn. The majority of the world is Rh positive. Okay? However, if you are pregnant and you're Rh negative, the chances of you having an Rh positive baby are astronomical. If you do have an Rh baby, the blood mixes between the mother and the baby. When Rh positive blood mixes with Rh negative blood, the mother will develop antibodies to the Rh positive factor. She will have a normal pregnancy. However, in the next pregnancy, she will have antibodies towards her baby. So it's important to understand that it is the second pregnancy that is the problem with an Rh negative mother. Because if the next pregnancy is an Rh positive baby, the Rh antibodies that have been developed from the first pregnancy will destroy the baby, the second baby. That's known as hemolytic disease of the newborn. There it is. Anti-Rh antibodies will kill the baby or have the baby born with severe malformations and not a long life. All right? You'll have a lecture on Thursday on sickle cell disease. I will not dwell on sickle cell disease. All right, that's chapter 19. Any questions on chapter 19?
I just have a question in general. When's it going to be uploaded to YouTube?
Probably this afternoon.
All right, perfect. Thank you.
Okay, no problem. I actually had a problem, and I think Anthony found that problem, and I'm glad, Anthony, you're here, are you not? You were here before.
Yes, I'm here.
Oh, okay. Yeah, you notified me that you couldn't find the lecture last week, and you were right. It wouldn't upload, but I did upload it. So now it's on. It's on.
Yes, that's all.
All right?
Thank you.
Okay, no problem. I appreciate you telling me that too. Okay, any other questions before we go to chapter 22? All right.
Nope, all good.
All right, here we go. We're now talking about the immune system and your lymphocytes. Remember, lymphoid stem cell, it's a separate stem cell from the myeloid stem cell, all right? But lymphocytes are white cells, but from a different host or mother, okay? All right. You need to know the difference between innate and adaptive immunity. Innate immunity is what you're born with. It's non-specific resistance. It's defense mechanisms that protect you against a wide range of invaders. The mucus in your mouth, the tears in your eyes, the saliva in your mouth, the enzymes in that saliva, the hydrochloric acid in your stomach, the bacteria in your colon, the pH of your urethra or your vagina. It's non-specific. Adaptive immunity, on the other hand, is very specific and uses specific lymphocytes to combat a particular invader. Very important to understand, all right? The lymphatic system is a series of structures and organs that contain lymphatic tissue or bone marrow, okay? And basically here it is. It starts off with lymphatic vessels, a one-way trip towards the heart. Unlike arteries and veins, it's only one way. The cardiovascular system is two ways. The lymphatic system is one way. Okay? There are lymph nodes in your body. There's red bone marrow in your body. There's also a spleen. There's also a thymus. And there also are ducts or ducts. The lymphatic system meets the cardiovascular system at the subclavian veins, the thoracic duct, which is on your left, and the right lymphatic duct, which is on your right, drops all of the lymph liquid into the subclavian vein, the left and the right. Okay? That's where your cardiovascular system meets your lymphatic system. The lymphatic system functions to drain interstitial fluid of waste. It transports dietary fats and it carries out immune responses. Okay? We talked about the vessels, we talked about all that stuff. It's a one-way system, All right? Here is the duct. The left lymphatic duct has its own name, the thoracic duct. The right lymphatic duct is the right lymphatic duct, okay? The right thoracic duct. All right? But look at this slide over here. The majority of the body is drained on the left side by the thoracic duct. Only the arm and the upper right side of the body and right side of the head is drained by the right lymphatic duct. Please know that. All right? And this is where the two meet. The ducts drop their lymph into the bloodstream at the subclavian veins. The primary lymphatic organs of the lymph system, where immune cells become immunocompetent, are bone marrow and thymus. When lymphocytes are in the red bone marrow and mature and become immunocompetent, they are called B cells. When lymphocytes in the thymus become immunocompetent and mature, they are called T cells. Your thymus is a large organ at birth. As you get older, it decreases in size and in strength. That is why when you reach my age, you are more susceptible to fractures, you're more susceptible to cancer, and you're more susceptible to infection. That's why we go to the front of the line to get the COVID-19 vaccination. And thank God I've gotten both of mine already, because not only am I an old person, I am a doctor. So I went to the front of the line. I was 1A. The secondary lymphatic organs are the lymph nodes, the spleen, and lymphatic nodules. You don't really have to know much about lymph nodes. That's not important. Just know where they are. Know when I say cervical lymph nodes, they're in your neck. And when I say inguinal lymph nodes, they're in your groin. Okay? Just know where they are. The spleen has many functions. One of them is to store platelets. And you're going to have a lecture on a disease called ITP on Thursday. And that student will tell you about the spleen involved in ITP. Okay? Very important. All right. There are also nodules all over your body that contain lymphocytes, and these lymphocytes are in a area called MALT, M-A-L-T, mucosa-associated lymphatic tissue. It's in your tonsils. It's in your gastrointestinal tract, where your small intestine is, to keep bacteria from getting into your GI tract. It's also in your lungs, also. MALT, M-A-L-T. All right? Innate immunity, you're born with it. You're born with skin. You're born with mucous membranes. You're born with internal defenses. Your skin is a mechanical defense. The tears, the saliva, the mucus, the cilia, your epiglottis, your urine, your bowel movements, your vomiting are all mechanical defenses to get rid of unwanted bacteria and dust. You also have chemical defenses. You have oil glands on your skin that produces sebum. You have lysozymes that kill bacteria, and you have gastric juices which contain hydrochloric acid that kill bacteria. Okay? You also have antimicrobial proteins. Antimicrobial proteins, some of them are interferons and some of them are cytokines. You're going to have a lecture on Thursday on cytokines. So you'll know what those antimicrobial proteins are. You have phagocytes. When you say the word phagocyte, you immediately should think of neutrophils and monocytes, the big eaters. There they are. Natural killer cells. You're born with lymphocytes that will kill things. Everyone has the same NK cells. You're born with a process called inflammation. It has 4 major characteristics: pain, swelling, heat, and redness. And you develop a fever to kill bacteria. Those are non-specific internal innate defenses. All right? What is phagocytosis? Phagocytosis is the destruction of a microbe. When a microbe is in the area of a cell, that cell puts out a chemical signal to the white cell to come down and get rid of this microbe. That's called chemotaxis. When the white cell comes, basically it's a neutrophil because it comes first. It will adhere to the microbe. Then it will ingest the microbe. Then it will digest the microbe. Then it will kill the microbe and then get rid of it. Those are the 5 steps in phagocytosis. Know them. These are your innate defenses. You can look at this at your leisure. I think I've gone through every one of them anyway, but look at that, okay? Non-specific. Here we are, interferons. Interferons are antimicrobial substances that protect uninfected host cells from a virus. The complement system, a system of 9 proteins that promotes phagocytosis and contributes to inflammation. NK cells, phagocytes, inflammation, and fever. Know what those do. Adaptive immunity. Adaptive immunity is specific and it has memory. It relies on not only killing things, but it also relies on the antigen antibody reaction. Antigens are substances recognized as foreign that provoke an immune response, and that provocation allows your body to either kill it or produce antibodies. And this is where lymphocytes are the most important white cell. All right? T cells mature in the thymus. B cells mature in the bone marrow. T cells kill things. They're known as CD8 cells. Helper T cells are known as CD4 cells. They accept a presentation of a foreign substance by a phagocyte. And they can either bring it to a B cell or a T cell. If they bring it to a T cell, that T cell destroys that microbe. This is called cell-mediated immunity. If this T cell brings it to a B cell, that B cell will produce antibodies. This is called antibody-mediated immunity. These B cells can become memory cells or active producers of antibodies, and those active producers of antibodies are called plasma cells. Plasma cells are B cells that are producing antibodies towards a specific antigen, cell-mediated, antibody-mediated. There can be parts of a bacteria, parts of a foreign protein that can make you produce antibodies. You don't have to have the entire microbe or the entire protein to be produced. And you can get what we call epitopes, parts of a huge bacteria, which is an antigen. This can be, you can develop antibodies towards the entire antigen, or you can develop antibodies against epitopes. Okay? I want you to think of this as the coronavirus. Parts of the coronavirus, one of the spikes, is now used in some of the development of the Pfizer and Moderna vaccinations. What they do is they take the mRNA spike and they make a vaccination. They make a vaccine. When this is injected into your body as an epitope, your body develops antibodies towards this mRNA epitope. And you become, after 2 shots, 88 to 98% immune to COVID-19. You will get a lecture on how vaccines work on Thursday by one of your classmates, okay? Antigen processing. We either kill it with T cells or we produce antibodies and kill it with B cells. Okay? I didn't really talk that much about major histocompatibility complexes, and obviously if I didn't talk about it, you'll have no questions on exam about it. Don't worry about MHC, that's not really important, okay? You're going to have a lecture on cytokines, how cytokines work and cytokine storm. on Thursday. I will not belabor this, but what I will belabor is interferons. Interferons are produced by virus-infected cells to inhibit viral replication in uninfected cells. It is a cytokine. It is a cytokine. What you should know about helper T cells, CD4 cells, if you remember my AIDS lecture, this is the cell that is studied in HIV infections. When the helper T cells, the CD4 cells, gets below 200, you now go from an HIV infection to full-blown AIDS. So that's a very important thing to understand. Okay? Very important thing to understand. Antibodies, immunoglobulins. You don't have to know the structures, but what you should know about the immunoglobulins is that these are the antibodies that are produced and to fight infection. The most abundant is IgG. IgG not only is the most abundant, it's the only one that crosses the placenta. IgA is the immunoglobulin that is found in fluids in your body. And all these fluids that contain IgA have an A in them. Sweat, tears, saliva, breast milk, gastrointestinal secretions. They all have an A in them. IgM. IgM is the first antibody to show up when you have an active infection. So if I drew antibodies, if you came into my emergency department with a rash on your body and I thought you had chickenpox and I wasn't sure, I would draw your blood and I would look for IgM antibodies for chickenpox. And if the IgM antibodies for chickenpox are high in your blood, then you have now been diagnosed with chickenpox. If you have IgG antibodies towards chickenpox, that means that you've had the infection in the past. In the past. This is present infection. This is post-infection. IgD is not important. Don't even study it. IgE is very important. It goes along with allergic reactions or parasites, just like eosinophils. It has an E in it. Eosinophils, IgE, allergic reactions, or worms. Very important. Okay? Complement is just a series of proteins that aid in phagocytosis and inflammation. You don't have to know anything else about it. That's all you have to know. That's all you have to know. All right? Here, IgM, the immunoglobulin that is high with active infection. IgG shows that you have memory to that infection. So they both are high. All right, that's important. So there are 4 types of acquiring adaptive immunity, and they are naturally acquired or artificially acquired. And those categories are broken down into active and passive immunity. Naturally acquired active immunity is you have the disease, you have COVID-19, you have chickenpox. Naturally acquired passive immunity is that your mother has given you the antibodies towards that disease. She can give you the antibodies transferred through the placenta with IgG, or she can give you the antibodies in IgA through the breast milk. Naturally acquired passive immunity. Your mother gives you the antibodies. Artificially acquired active immunity is when you get the vaccination. You get the vaccination for COVID, for chickenpox, for measles. You get the virus or a part of that virus in a vaccination. Actively acquired passive immunity is when I give you the antibodies. And this is a big treatment in people who are COVID positive, who are at risk, elderly, have disease. They go to the hospital and they get an hour-long injection of the immunoglobulins for COVID. All right? So know these four categories. Know these four categories. All right? We talked about immunological disease and self-tolerance. This is a good review slide. This is a good review slide. Remember that macrophages are known as APCs, antigen-presenting cells. Remember, through phagocytosis, neutrophils and monocytes get the antigen inside. Before they get rid of it, they present it to cytotoxic T cells or helper T cells. If they present it to cytotoxic T cells, CD8 cells, these cells will kill the antigen. If they bring it to a helper T cell will cooperate and bring it to a B cell, which will produce antibodies, becoming a plasma cell, and through antibodies, that antigen is destroyed. Okay? As you get older, as we said before, you lose your ability to produce more antibodies. You have a decreased response to vaccines. That's why the flu shot that's given every year, there's a specific flu shot for the elderly. It's called Fluzone. And that's the one that the elderly gets. The regular population gets the routine flu shot, where the elderly gets a stronger vaccination. All right? We talked about AIDS. Know about helper T cells or CD4 cells. We talked about allergies. Know about epinephrine and Benadryl. And we talked about autoimmune diseases. All right? I will now unrecord. Let's see what I can do here.